Hello everyone, my name is Andres Lopez and I'm a second-year student in Dr. Stein's group. Today I will talk about the research about the effect of microbial phytase on standardized total tract digestibility on phosphorus in feed phosphates by growing pigs. As an introduction, phosphorus is the second most abundant mineral in the body, and it can be delivered to pigs in two different ways. First, plant ingredients which have a low digestibility of phosphorus, and this is due to the presence of a molecule called phytate. Phytate is the natural storage of phosphorus in the plants, and in order to be broken down, we need an enzyme called phytase. But this phytase does not occur naturally in the gastrointestinal tract of the pig. Therefore, this enzyme is also supplemented in diets. And the second way is using inorganic sources of phosphorus with a high digestibility of phosphorus, and in this case, the ingredients that we can use are feed phosphates, such as monocalcium phosphate, monosodium phosphate, and magnesium phosphate. But when we analyze a molecule of phytate, we can also see how these will naturally interact with other minerals, such as calcium, binding up to 6 molecules of calcium. However, recent research has indicated that the use of phytase in diets containing calcium carbonate will increase the digestibility of calcium. Having this in mind, we conduct an experiment with the objective of test the hypothesis that microbial phytase could also increase the digestibility of phosphorus in feed phosphates. For this experiment, we use 170 pigs with an average body weight of 15.56 kilograms. And in this case, we formulate 3 basal diets with the inclusion of 0, 500, and 4,000 phytase units. We include one of the 3 different sources of feed phosphates that we use in this experiment, which are MCP, MSP, and MGP to the basal diets, and therefore we include 12 diets in this experiment. Also, a phosphorus-free diet for a total of 13 diets. The pigs in this experiment were allotted into these 13 diets, and they were housed individually in metabolism crates with free access to water and being fed twice a day. In this experiment, we had an adaptation period from day 1 to day 5, and then an undigestible marker was fed to start a collection period on the day 6. This collection period will go until day 10, where they were fed a second undigestible marker, and then they will be collected until the second undigestible marker pass, which was in average until day 12. First, for the nutrient composition of the ingredients, I would like to set my slide, and in this case, MCP will be represented in blue, MSP will be represented in orange, and MGP will be represented in yellow. For the concentration of phosphorus and calcium, the results were in agreement with reported data, but in the case of magnesium phosphate, the concentration of phosphorus was lower than expected. In the case of corn and soybean meal, the concentration of phosphorus, calcium, and phytate were also in agreement with expected values. Moving into the results of digestibility of phosphorus, first I would like to set up my slide. You will find the basal diets represented in green, the MCP diets represented in blue, the MSP diets represented in orange, and MGP diets represented in yellow. Also, the lightest color will represent the inclusion of 0 FTUs, the darkest color will represent the inclusion of 4,000 units of phytase, and the color in the middle will be representing the 500 units of phytase. And as we were expecting, the digestibility of phosphorus in basal diets increased with the inclusion of phytase, from around 45% to almost 90%. And if we analyze the results from MCP diets, what we can observe is an increase in the digestibility as we increase the inclusion of phytase, but we can also observe a difference between the basal diet with 0 phytase and the MCP diet with 0 phytase, which indicates the increase in the available phosphorus when we include a source of inorganic phosphorus. From MSP, we observe the same results, an increase in the digestibility as we increase the concentration of phytase, and the same results were observed for MGP. However, the results from MGP appear to be lower. However, this is the digestibility of phosphorus in the diets, and the objective of this experiment was to test if the digestibility of phosphorus in the ingredient is different. For that reason, we use the difference procedure to analyze the digestibility of phosphorus in used ingredients. And in this case, blue will represent again the MCP, orange will represent MSP, and yellow will represent MGP. And what we can observe is that there was no difference in the digestibility of phosphorus in MCP, MSP, or MGP. The only difference that we can observe here is that MGP had a lower digestibility if it's compared with MCP or MSP. So, moving into the conclusions, the digestibility of phosphorus in feed phosphates was not affected by the inclusion of phytase in the diets. The effect that we observe in the diets is the effect of phytase on the phosphorus present in corn and soybean meal, which was expected. Also, we can observe that the digestibility of phosphorus or STTD of phosphorus in magnesium phosphate was lower than MCP and MSP. With that, I would like to acknowledge Yara for the financial support to conduct this experiment. And then I would like to say thank you to the Monogastric Nutrition Laboratory of Dr. Hans Stein. And if you are interested in nutrition, please follow us in our social media. Thank you for listening.